One of the schemes that we support and has been very successful have been the Student Research Bursaries. So these are actually fairly modest investments, but we've found that the payback has been tremendous actually. So we're really keen to support younger researchers and pump prime their research projects. So we're going to have two of those presentations. The way we're going to run this session is we'll first of all have the award presented to the recipients, and then the students will give short presentations about their research plans, and tell you a little bit more. So the first presentation will be from Alisha Sharma, who's from the University of Southampton, and she will be awarded the Sir Julian Paget Student Bursary Award. And so I'd just also like to invite Sir Henry Paget to join us and to make this presentation. So Sir Henry, as many of you will know, is a patron of the Pagets Association, and our most active patron. Is a tremendous supporter, and we really appreciate the efforts he makes to work with us. Sir Henry is the great-great-grandson of Sir James Paget, my mistake, who gave his name to this condition, and the award is made in memory of his late father, Sir Julian Paget. So this is the Sir Julian Paget Student Research Bursary. So I'll ask Sir Henry to make the presentation to our student, and then I'll ask our student to, afterwards, Alisha will tell us a little bit about her research at Southampton, I think. So thank you. We're going to use this as a photo opportunity as well, so there'll be a slight pause as well.
So thanks for that introduction. I'm Alisha and I'm from Southampton. I recently got my bachelor's in biochemistry and I'm now doing a PhD in bioengineering. So I'm going to talk to you a bit of what my project is going to look at, my future project of whether blood vessels have an involvement in Paget's disease. So as we've seen from today, and as you already know, there's so many problems in Paget's disease. And one of the problems is the poor bone quality. So in healthy bone, you have structured bone, and this is what it kind of looks like here, and we call that lamellar bone. And in Paget's disease, you actually have weaker bone. This bone that's actually formed, this excess bone, is actually weak, and it kind of looks like this. As you can see, it's far less structured in that far picture compared to what a healthier bone should look like. And because of this, you can have problems like fractures. And like Mark was saying, these fractures can actually be dealt with by having surgery. But there's problems with surgery, as we've heard. So because of the deformity itself, you have to be able to treat the deformity, and that's kind of difficult to get around. The weak bone, like I've just said, the woven bone poses problems. And like Mark said, you have this excessive bleeding in surgery. So sometimes the blood has to be pumped out of the bones itself, and this causes a problem. So what are my research questions addressing? Well, could the blood vessels be involved in Paget's disease? So in bone, it's actually very highly vascularized. A lot of blood supply is for the bone because a lot of bone cells need nutrients so that they can function properly. So I'm wondering, could the blood vessels actually have an underlying cause in the disease? And then are the blood vessels different for males and females? So our actual lab group back in Southampton have shown that blood vessels differ in males and females. So I kind of want to see if this was the case in Paget's patients also. So I've done my PhD involves looking at Paget's disease as well, and I've firstly kind of looked at bone quality. So I've looked at bone shape specifically, and looked at the skull and leg bones. So what I did to do this was actually using something called micro-CT. So I'm sure you've heard of CT scanning, or maybe just heard the name, or if you haven't, micro-CT is very similar, but just much more powerful. So what it involves is actually using high energy X-rays, and you kind of shoot this at your sample, and then this is detected by this source at the end. So if you can see, it's quite small on the screen, but that sample is actually rotating. So essentially what you're doing is actually taking pictures of your bone at every single angle so that you can collate all these images, add them together and produce a 3D model of the sample that you've got. And you can make quantitative measurements. And I've done that for my leg bones and skulls. So moving on from that, I can actually look at the bone strength. And I've done this with leg bones. And I've actually produced a bending test to do this. So what do I mean by bending test? Well, I've created this 4-point bending model. So that means you actually apply force to the bone and you kind of you cause it to fracture. And from this you can record the amount of force required to break that bone. So this is just a little kind of video of it. So you apply the force and eventually it causes that bone to fracture. So specifically, what's good about this model is it actually allows you to define the weakest point of that bone. And what I want to do is actually look, where is this bone weak? Where is the weakest point of this bone? And what we found is that it seemed to be in this kind of distal region in the samples we were looking at. And interestingly, with the changes in bone shape and this experiment, we kind of noticed it was always in this distal region. But very interestingly, from all this research so far, is that we've noticed that there's sex differences. For example, in the skulls, the males seem to be more affected in this case. And in the femurs and the leg bones, there were different changes. Males had changes in, for example, cross-sectional area, whereas females had more changes in thickness and different parameters. And I don't know, maybe this could be due to sex hormones. This really needs to be looked into, I think. There was a case by Hadaway, actually, who was saying that females actually have more monostotic cases, where normally one limb is affected, and males seem to have more polyostotic cases. So maybe this needs to be looked into more. So what I'm going to use the bursary for now is actually my next step. So I don't know if you've ever seen this before. Kind of looks like something from the Avengers. It made me feel like I was in the Avengers when I was there. So what it is essentially is somewhere I can do really, really powerful micro-CT. So blood vessels are actually really deeply embedded within your bone, and that makes it hugely difficult to be able to study. So I actually got the opportunity to go to synchrotron, which is the diamond light source synchrotron, and that's in Oxford. There's also one in Switzerland that I've been to, and I've done some scans there, and this allows me to be able to see the blood vessels within bone. So if I talk about that in a bit more detail, actually, let me explain to you what this means. So they actually accelerate electrons and around this ring here as fast as they can. And what happens is they kind of pull off the beam. The beam comes off in these little compartments, and you're actually in this little compartment, and you can use that beam to scan your samples. So what I mean by this is if I take my bone and I'm looking at a small section of it, so you'll do multiple scans looking at small sections, I will generate images like this. Now this is what the inside of your bone looks like. Not sure if you thought that's what it looked like. That is what it looks like. So you can see it's full of holes, and that's normal. The tiny holes actually are called osteocyte lacunae, and that's where your osteocytes, which are a bone cell type, reside. And then you have these slightly bigger holes you can see, and that's where your blood vessels reside. So doing this, you can actually extract just the pores, so remove all the bone away, so you're just looking at those holes, and then create a 3D map looking at this, so you can look at all the osteocytes together and the blood vessels. And then through this, you can just isolate the blood vessels. So now I can actually quantify these blood vessels and assess how they make a difference in Paget's. So what I'm predicting, because I'm about to do this project, is after the processing, that the Pagetic bone actually has more vasculature, and compared to the healthy bone, it's more vascularized. And if this is the actual case, then maybe blood supply should be the therapeutic target. Maybe we should be targeting the blood supply as a future treatment. And like I said earlier, could these treatments be targeted for males and females? Because we're seeing very different sex differences between for different things, bone shape, bone geometry, and maybe the vasculature. So maybe that's the way forward. I just want to thank everyone for listening to my talk, and all the collaborators I've had so far. And I especially want to thank the Paget Association for funding my project. Thank you.
This is the Alan Reid Student Bursary Award, and again, some of the members may recall that Alan has been associated, or was associated, with the Paget's disease Paget's Association for many years, and we had a long relationship with Alan, and provided him with support when he was affected. He was a patient. And we have a bursary in his name that we will be awarding now. So the second bursary will be awarded to Nicole Ball from the University of East Anglia, and I'm going to call on Professor Stephen Tuck, who's our Vice Chair. Stephen, if I could invite Nicole as well to come for her award and then to sing for her a supper as well. I say Nicole's from the University of East Anglia, and I'll say more about her presentation in 1 second when we've got the photos. We're going to get molecular, and Nicole's going to tell us about an important protein in Paget's disease called sequestosome, and I'm particularly pleased because I'm also talking about sequestosome afterwards, and so my introduction hopefully will be covered and I'll have less work to do, so thank you.
Hi everyone, I'm Nicole and I've come from the University of East Anglia today, and I'd just like to cover what my project is going to involve, which is looking at a novel role of sequestosome 1 in Paget's disease as well as Paget's associated osteosarcoma. So firstly, as was mentioned, I'd quite like to dedicate my talk to Alan. Alan suffered from Paget's disease for a number of years before sadly passing away from Paget's associated osteosarcoma in 2014, so I would like to dedicate my talk to him today. So firstly, I'd like to cover a little bit of background on bone remodelling, which we've heard a bit about today. So in bone remodelling, the two cells I'm going to be talking about today are osteoclasts and osteoblasts. So firstly, involved in bone resorption, which is the degradation of bone, we have the osteoclast bone cells. And then secondly, in the bone formation, we have osteoblasts, which lay down collagen, which is mineralised to form the new bone. Now this cycle of osteoclast degradation of bone and osteoblast formation of new bone is a continuous cycle which happens throughout our lifetime. And it remains relatively in balance, the activity between these two cells in healthy adults. Now in Paget's disease of the bone, as many of you all know, it's the second most common metabolic bone disorder, and it occurs when this cycle of bone remodelling is abnormal, and we get an increase in the osteoclast cell activity. So there is an increase in the breakdown of bone, and this is followed by a stimulation of osteoblast activity. So the rate at which new bone is being formed is increased, and because new bone is being, the rate of new bone is being formed at such a fast rate, the new bone that is formed is fragile and is prone to fracture. And the structure is very different, as we have heard about today. So the top two pictures of the femur and the tibia are in normal bone, and then the bottom two pictures are from progetic bone. And you can see the thickening of the bone and how porous the structure is. So a rare complication of Paget's disease is Paget's associated osteosarcoma. So in normal bone remodelling, we have the osteoclast activity, which degrades the bone. This stimulates the osteoblasts, which then move to form new bone in the sites of osteoclast activity. However, in Paget's disease, the activity of osteoclast is significantly increased, so there is more degradation of bone. So the activity of osteoblasts is also increased, so the formation of new bone is happening at a much faster rate, which I mentioned. Now, in a very rare cases, the osteoblasts can go through some malignant changes and become cancerous cells, and this is where we get the formation of Paget's associated osteosarcoma. Now, I should stress that this is a very rare complication affecting about 1 in 1,000 Paget's disease patients. So what causes Paget's disease? So the exact cause of Paget's is still unknown, however it's thought to be a multifactorial disorder. And what this means is that both genetic and environmental factors are playing a role in the development of the disease. So in familial Paget's disease, researchers identified that there are several genes which predispose patients to developing Paget's. And one of these genes is known as sequestosome 1. And as I say, it's quite common in the hereditary form of Paget's disease. So where does sequestosome 1 come from? How is it formed? So within the cell we have the DNA, and within the DNA we have the genes for different, which produce different proteins. So one of the genes is sequestosome 1. And we can think of the DNA within the cell as the blueprints of the cell. The information in the DNA is copied into the RNA, and so the information for that sequestosome 1 gene is also copied over into the RNA. And we can think of the RNA as the instructions of the cell to produce different proteins. So from here, the sequestosome 1 protein is produced from that information. And you can think of the protein as the tools for the cell to perform different functions. So one of the functions, as I've mentioned, is, of sequestosome 1 is the bone remodeling, so it can leave the cell and stimulate bone remodeling. However, there has been some evidence to suggest that sequestosome 1 is able to return to the cell and interact with different RNAs. And because of this, it would be known as a RNA binding protein. And this is one of the things I'm going to be investigating with my student bursary. So this diagram, just to repeat what I've just said, is for the normal production of sequestosome 1. So we have the sequestosome 1 gene within the DNA, copied into the RNA to produce the sequestosome 1 protein. However, in Paget's disease, there is some a sort of mutation which is causing damage to that sequestosome 1 gene. Because there is damage in that gene in the DNA, when it is copied into the RNA, this damage is also copied over, so that the instructions for the protein production are now abnormal, and this produces an abnormal sequestosome 1 protein. As you can see from this diagram, the sequestosome 1 protein that is produced in Paget's disease is truncated, meaning that the tail end of the sequestosome 1 is missing. And this tail end of the sequestosome 1 protein is really important that it is able to be labelled for degradation. So in Paget's disease, because this tail end is missing, it is perpetually active, which is continually stimulating the bone remodeling cycle. And this is where we get Paget's disease. So how does the cell recognise that there is an abnormality in the RNA information? So there are other forms of RNAs called microRNAs, which are target for specific RNAs, and they're able to silence that RNA. So evidence from our lab has identified that a specific microRNA called miR16 is able to recognise that sequestosome 1 is faulty in the RNA. The miR16 is able to silence this RNA and stop the production of the protein being produced. So from this diagram which we saw earlier, you can see the faulty gene of sequestosome 1 in the DNA copied to the RNA. Now this miR16 is able to recognise that that RNA has got the fault, and you can think of the miR16 as a delete button on this process. So it interacts with the RNA and prevents this sequestosome 1 being produced. Now we may think of this as a positive process in Paget's disease, as obviously if this abnormal protein is what's causing the accelerated bone remodeling, you'd think that the miR16 expression would be a really good thing. However, this isn't always the case, and I'm going to go into that now. So just to recap on sequestosome 1, it is involved in accelerating the process of bone turnover, and in Paget's disease, when it is mutated, this is accelerating the bone remodeling. One other important role of sequestosome 1 is protecting the DNA from damage. This is just a surface representation image from some of Professor Robert Layfield's work, and it just shows that surface representation of the missing region of sequestosome 1, which I mentioned before, which is needed for the degradation. So a couple of questions that I'll be covering in my research. So does the overproduction of sequestosome 1 once it's mutated have a protective effect against cancer? Because if sequestosome 1 is protecting the DNA, this will prevent malignant transformations in some ways. However, does the silencing of sequestosome 1 by miR16 actually lead to the Paget's development? So if miR16 is acting in such a way by silencing sequestosome 1 altogether to prevent the abnormal protein, does this lead to the DNA being unprotected from damage, and which is causing a Paget-associated osteosarcoma? So we like to think that within the cell, this is what we're hypothesizing, that there is a battle going on between the protein, sequestosome 1, and the mRNA miR16. So just to summarize, the sequestosome 1 is mutated in the Paget's disease, is really highly expressed in Paget's disease as it's perpetually active, and this allowed to proliferate and cause focally increased disorganized bone remodeling. Now the miR16 is able to turn off sequestosome 1. Interestingly, it's really highly expressed in Paget-associated osteosarcoma, however, it is barely seen in Paget's disease. So there is a regulatory effect of miR16 on sequestosome 1, however, it isn't seen in Paget's disease. So the sequestosome 1 is silenced, meaning that DNA can't be protected from damage, and this is what we think is lead to the Paget-associated osteosarcoma. So the method that I'm going to be using to investigate these RNA and protein interactions of sequestosome 1 is called RIP-seq. And I'm just going to summarise just the basics of this methodology to you all now. So I'm going to be collecting blood samples from Paget's disease patients as well as controls. I'll be breaking down these cells in order to release the RNA, and it will be interacting with proteins such as sequestosome 1. I'm then going to be using a technique called immunoprecipitation. Now this involves using an antibody which acts like a tag for sequester zone 1. So this antibody is specific for that protein. I'll introduce it into the cells and it will be able to bind to sequester zone 1. Now previously to this I should have mentioned that once I break down the cells, the sequester zone 1 will be locked onto the interacting RNA using UV light, and it forms strong bonds between the interacting protein and RNA. So once I introduce this antibody specific for sequester zone 1, I then form a complex. The antibody binds to sequester zone 1, which is interacting with its RNA. I can then wash off any unbound material in the sample so that I'm left with just this complex of the antibody, the sequester zone 1, and whichever RNAs it has been interacting with. I can then wash off the antibody in sequester zone 1, and this will leave just the RNA that has been interacting with our protein of interest. This RNA is isolated, and due to sequencing techniques, we'll be able to identify which RNA was interacting with sequester zone 1. So just to recap, the student bursary, which I'm going to be investigating, is I'm going to be looking at the RIP-seq technique to look at the role of sequester zone 1 in Paget's disease, as well as Paget-associated osteosarcoma. As recent evidence has suggested that sequester zone 1 is acting as an RNA binding protein, I'm looking into which RNAs are interacting with this protein in Paget's as well as Paget-associated osteosarcoma. So we should see that it is interacting with the miR-16, but there may be other RNAs which are interacting with sequester zone 1. We hope to provide a better understanding of sequester zone 1 in this disease, which may lead to future therapeutic treatments if we can intervene with these different interactions. So I'd just like to thank my supervisors at University of East Anglia, so Dr. Darrell Green and Professor Bill Fraser, as well as the Paget's Associated Association and the Reed family for setting up my Student Research Bursary. So thank you very much, and if anyone's got any questions, talk to me afterwards. Thank you.